viewers, this is Mr. Ogeno. Welcome to today's lesson, which I want us to talk about the genetic coding. Now, the first thing that we already know is that if you look at DNA is a double helix structure which can replicate itself so that it can give us the information about itself to form materials that we need to make up new cells. We need to make up the whole organisms. That is what the DNA is all about. Now, if that is the thing that we already know, we know that the DNA therefore can be linked to the control of the protein synthesis in an organism. So if that is what we talk about, the building up of the organisms, the next thing that we want to know that proteins are made up of amino acids. That is what we know the proteins are. So we can say that proteins are made of, they are made up of amino acid. Now we know very well that using the DNA code, we have got only 20 natural, we have got 20 naturally occurring amino acids. So we have got 20 naturally occurring amino acids. This is what we have. We have got 20 occurring amino acids. Now the next thing that we know that we have to ask ourselves, how is this particular genes formed? How is the amino acids or the proteins normally formed? Now we know that we have got the base sequence of the amino acids, like we have got the 4 bases that is found in the amino acids. The other part of the DNA are constant, like we know that we have got the pentose sugar, we have got the phosphate. So the only thing that we have here that can be varying based on how they are the nitrogenous bases. So we have got, remember, we have got the thiamine, we have got guanine, we have got cytosine, and then we have got the adenine. So you see, these are nitrogenous bases and they are 4. So they are the one that are at least different here. Thiamine different from guanine, from cytosine and adenine. But the remaining part of component of the DNA are the same, like the pentose sugar, like I've said, and the phosphate sugar. So when we want to understand how this particular protein is synthesized in a cell, then much of the work will be done on understanding how this particular nitrogenous bases are going to exist in a specific way. Now we know that this particular base therefore can be coded in a specific way to give us a specific protein. Now this particular protein, once that one has been done, the coding has happened within the DNA, which is found in the nucleus, the protein synthesis is found in the ribosome. That is what happens, that the information that leads to the protein synthesis within the nucleus of the cell, but the actual protein synthesis is done in the ribosome. Ribosome is an organelle that is found outside the nucleus. So there must be that passage of the information. So we know that the bases, how we have them, the information will be transferred from the DNA to the ribosome. Specifically, this one is done by an RNA. So they must be having a specific code that lifts that information, and then it has to be transferred here. We know very well, and we have learned that the RNA itself, RNA also has got nitrogenous bases. The specific one that they have is we have got the guanine, cytosine, adenine, and uracil. So it means that in terms of the coding, the G will code with a C, and then in RNA and the DNA, the A in the DNA, the A will be coding with a U or a T based on where it is found. So this is how they are. A normally code with a U or a T based on, are we talking about the the DNA or RNA, because you know that the T is only existing in the DNA, but the U is also only existing in the RNA. So that is how the coding will be done. So it is now therefore called upon the scientists to realize what normally is the genetic code. We have to know what is now coding, what is now this particular coding, so that we have got the information being relayed very well. What is the genetic code? Maybe we may ask ourselves this particular question. Now we can define that a genetic code is a sequence of the base on a DNA. We can say that the genetic coding is a sequence of the base on DNA, on the DNA molecule, which codes for specific amino acid, which code for specific amino acid. That is how we can define it. So this is just a part of the DNA molecules. We have got the polypeptide, the polymer of the amino acids when they are arranged in a very long chain. So that particular pattern that is coming, that is based on the nitrogenous bases that exist in that particular pattern, is the one to describe or to stand for a specific amino acid to be synthesized. So we can therefore agree that in a sequence of the nitrogen in a DNA molecule, that particular small section of it will be formed, that is due to the arrangement of the nitrogenous bases, will be giving us the base sequence of that particular DNA. So there is a specific code that is done to give us a molecule that will be synthesized within a DNA or in the protein synthesis that happens in the ribosome. So that is what we are calling the DNA coding. Now in the sequence of this particular coding, we know that normally we already have got how many molecules do we have? How many nitrogenous bases do we have? Now we already have got 4 known nitrogenous bases in the DNA. We have got the T, G, C, and A. These are the nitrogenous bases that we have. So scientists have come up with theories to suggest how these ones can normally work for us. So if you only have got the 4 nitrogenous bases, that is what we have. So if this particular, the possible arrangement of these particular codes, how are they? So if, for example, we say that we have got the 2 nitrogenous bases giving us the code, what will be the total number of the genes that we are likely to have? So if that is the case, then if I have got 4 having the 2, so I can have 4 by 2. These are the possible coding that we can have to give us the amino acids that we are talking about. So these ones will be giving us only 16 amino acids. This is less than the 20 that is required if you are together. Remember that there are 20 natural occurring amino acids. So if any case the sequence was to be in a manner that we are having them only the 2 bases that are pairing, so we can be giving that if they have got the total number of nitrogenous bases are 4, and then they can only code in a format of 2, we will be having 4 by 4, and then we are going to get 16 nitrogenous bases. So maybe this is the possible nitrogenous bases that we are having. So you see, this suggestion will not add up to what we are supposed to expect there. So we have to think, what is the possible way that that these particular genes can code? Now suppose they are coding, if you have got the triplet codes, for example, the nitrogenous bases can be in the form of 4 by 4 by 4. You can see this one is giving us 64, which is way already ahead of that. So it means that there is a specific pattern of the coding of this particular protein giving us a very good sequence. So maybe this one can give us an accurate information, because towards the end we can say that if we have got the overlapping one, it can be giving us the end of the first sequence, and it can give us the beginning of the new sequence. So that is how the coding of a DNA molecule is. So up to this particular point here, we must realize that we must know what the code, the genetic code is. That is the most important thing that we can know up to this particular end. And we know that it is the sequence of the base on a DNA molecule which code for a specific protein. So that is an information that is very important for us to really understand up to that particular end. Now, if we talk about, if we continue understanding this, we may realize that for this particular coding here to help us in the protein synthesis, we have got two specific molecules which we have talked about. So we have talked about DNA molecule, and then there is an RNA molecule. These particular two molecules are really important in the protein synthesis. Now we know, and we have really known that from the very beginning, that the DNA is entirely found within the nucleus of a cell. So every cell has got a nucleus, and they are restricted there. That is the first thing that is there. And then the next thing that comes in is that where is this particular protein synthesized? The protein synthesis is done in the ribosomes. Ribosomes is out of the DNA. So the next thing that will always come to us or to our attention is, how is this particular information relayed? Though we will look at that in later lessons, but we have to ask that question now. So the first question that we may ask ourselves up to this particular moment, we can say that there is already a code for the DNA. So there is that particular specific coding of the DNA. And then there is also the code for the RNA. Those two things here, they have to code. So this particular coding here will be having a pair. So if we have got the DNA codes here, the DNA code, there is that particular code that must complement it. So there is that complementary code for the RNA for the information to be passed. And therefore this one will be giving us the complementary strand of the DNA. The complementary strand of the DNA molecules will therefore be giving us a shorter thing that we are calling messenger RNA. Messenger RNA, which we normally write as mRNA. Now this is the RNA that can pick the information from the DNA within the nucleus and then move with it. So we have got various RNA, as we are going to see later on. There are three types of RNA that we'll be looking at, and their position and what they should be doing at what position. So at this particular moment, we have to know that the messenger RNA is the one that is having a code that complements. It is the complementary strand of the DNA that is going to link us in the protein synthesis. So that is what is basically we are understanding at that particular point. So always in this particular strand, we have to know that the DNA, we have known how they normally are combined. So the G here will always be combining with the C in the RNA. And the C also on the other way will be combining with that. Then when we come to this, the A here will be saying that the T is, the T here is going to combine with an A here, or an A with a U, because we know that this particular T is missing in the RNA. What we have in its place is a U. So if any case we have got an A here, we are expecting here to be a U. So that is how they are. So there is how this particular three will be interchanging based on the position where they are found. So that is the first thing that we should be remembering, how this particular coding is done in this particular molecules. So that is very, very much important for us to really understand up to this particular moment. Now, if this is the case that we have known that there is a code, how these particular molecules normally code for a specific protein or specific protein molecules, how will this particular code normally work out for us? How is the coding working out for us? So maybe we may ask ourselves that question and we can find out the meaning of that. Now for this, there was already a study that was done when we were talking about the gut bacteria. That is the E. coli that was used in the study on giving us on how these particular codes are done. Now we know that there is normally there is a specific percentage of the human protein molecule or the bases here that do not have got a specific thing. Like for example, we know that we have got the study revealing that out of 98%, out of the 98% of the codes here do not code for a specific thing. So that is how the DNA molecules are. So they are like non-coding. So the human DNA, 98% of them are non-coding. So we are only left with around 2% to be the one that is giving a specific code for the human protein. Like for example, we know that if we have got we can talk of the first particular in the polypeptide here. The first particular protein that we can code for is we gave it a name called methionine. So this is the first code that is done, that is given by a TCA. So these are the triplet nitrogenous base that is coding for this. So there are specific known proteins that only a specific percentage of the section of the DNA can code for them. Then the remaining bigger percentage is referred to as non-codon. So we want to look at it this way. This particular code that we have here, are they overlapping or they are non-overlapping? Can the genes overlap? So I want us to understand what is the meaning of the genes overlapping, or are they degenerative? That is, are they something that is indefinite? So how are this particular coding done? So maybe at this particular point we can come up with a table that is going to help us come out with the genes, how they are coded, and so that we can see how these particular molecules code out. Now we already know that the genetic code is not just a triplet code, but it is also a non-overlapping code and also degenerated code. Now I want us to try and understand what we mean by non-overlapping code. I want to use an example here that can help us to break down what we are talking about. Assuming that we have got an RNA sequence that is having this particular nitrogenous bases, that is I'm saying this is an RNA sequence with the following molecules, nitrogenous bases in them, GC. Now when we talk about non-overlapping, let us look at this and then we understand. We know that there is always a triple base that give us a code for a specific protein synthesis. So assuming that this is the sequence that I have in a polypeptide strand, if I take the first three, that one is a code for a specific protein. Now in the table that I have here is what we are using. I have given the code for every protein that is possibly synthesized. Now from here up to here, this is a DNA strand that I was using. You can see that I have got adenine, guanine, thiamine, and cytosine. So there is a thiamine here, there is a adenine, guanine, thiamine, cytosine. So this one here is a DNA strand. That is where I took it. Now this one I took from RNA strand. That is where there is uracil, cytosine, adenine, guanine, and that is what I'm trying to indicate here. So every protein here has got a code. So if, for example, remember that this particular RNA molecule here or strand is a complementary strand for this. And what does that one show? Like I want to just explain what we mean by a complementary strand. Let us look at every individual protein that is synthesized. Like for example, I can talk of phenylalanine here is a protein. This is its code in an RNA. Let us look at phenylalanine in the DNA. So if I have got here, there is an adenine. This one is responsible for phenylalanine. Now look at it here. A normally pair with U, A with a U, and A with a U. So this is a strand in RNA that is complementary to this. So how do we talk about overlapping, that the gene is non-overlapping? So assuming that this is what we have, if I have got UUU, which code is that? Now in our table here, that is phenylalanine. That is a possible protein that can come out of that. We can say that if any case it is overlapping, the next one means that it is from here up to there. That is UUA. Which protein will be that? That will be a leucine in the RNA strand. So that is what we are getting from that. If we continue with the process, I can have that is UAG. Where is our UAG? In this particular strand, we don't have it. We have got UUG. So that is what we are having, like an overlapping. So UAG. So it is going to UAG will be giving us a stop code here. That is what we have. We can continue with an AGC. An AGC. An AGC. An AGC. An AGC here, seranine. So if any case that this particular code is really overlapping, we are going to have very many proteins that can be synthesized within a simple sequence. So this one here is like it's going to give us it's like making the work very easy here. Like we can be having very many protein in a simple sequence, which is not the case. That is not true. And there can be a problem whenever we have got this kind of result being there. Like for example, assuming that if in the event that there is a problem, like in the case of mutation, what will be there? Let us assume that this particular molecule here, I mean this nitrogenous base here, that one is affected in terms of mutation. How many proteins will be affected here? Now if you look at it keenly, there are possible 3 proteins or 3 amino acids are completely affected. So these ones can be bringing a problem to individual organism. So this one here tells us that the gene is very risky, and it is not in order for the genes to overlap here, the bases to be overlapping. So we find that from here there are non-overlapping codes. So the first 3 here, they will be independent in the sequence of the DNA strand. So each one of them will be representing amino acids on its own. It's a code on its own, and then the next one will follow. So out of this we will be getting codes like the possible codes that we'll be getting there is U, that is a code, and then there are 2 AGC. That is the codes that we have. So out of this sequence that we have here, so I have got 2 clear amino acids that can be synthesized from this parallel coding. I can be synthesizing phenylalanine, and I can also be synthesizing AGC, which was AGC seranine. That is what those are the nitrogenous bases that I can be getting out of this. So this is why we say that the sequence here is non-overlapping codes. So that is one thing that we should be here. So this particular table here is clearly giving us out the type of the molecules that we have that can be synthesized and their codes that we have from them, from both the DNA and the RNA. That is one thing that we really have to know. Now, in the event that we are going to talk about the degenerated codes, this one simply means that they're redundant. So if any case we have got these particular codes here, the way we have them on the table here, what are we talking about when we talk about that the genes are degenerated? So if we look at these particular molecules, that the way they appear on our table here, we can really see that these particular genes here, one thing that control, if you follow the table very well, the gene here, is the first two letters of the base. That is what control the code. That is what controls the code of what is going to be formed. Like for example, I want us to look at this one here. If we look at the first thing here, the leucine here, we have got G and A. The last letter is an A. G and A, the last letter is a G. G-A-T, the last letter is a T. G-A-C, the last letter is C. But all of these particular codes are responsible for this particular amino acid. That is what we have. So the first two letters are the most important particular bases, the bases that are responsible for a specific protein synthesis. If you follow it keenly, you will realize that that is what is really important. If any case that these particular letters were each one of them give us a specific amino acid, then it could be giving us indefinite amino acid that could be synthesized, that will be bringing in the redundancy that we are talking about in this parallel synthesis. So the key thing is that the protein is synthesized in a specific code. That code is controlled by the first two nitrogenous bases in a sequence or in a code of 3 triplets. So that is how the genes are always responsible for the formation of a specific protein. So that is one thing that I really want us to understand. Now the next time that we meet, we want us to talk about the DNA and the protein synthesis. What is the role of the DNA in the protein synthesis? So we'll be looking at the different types of the RNA that we have. Like I've talked about one RNA here that is coding with this particular DNA in the nucleus, that is the messenger RNA. So we are going to see what the messenger RNA does, and the remaining two RNA, what do they do and how are they responsible for the protein synthesis. Thank you very much, and let us meet next time.